حي على الصلاة حي على الصلاة حي على الصلاة حي على الصلاة.
Okay, so this gives a sense of my journey. Not this is a tardigrade, which is a very interesting little animal, really, that I've started
So,
observing on a microscope when I was a kid, and actually, I mean, it's really the observation. It wasn't as good as an image as this one, but it's really the observation of this little creature that got me really excited about biology. And I did study medicine in Milan, where I'm from, and I had a fourth year of medicine in Stockholm as an exchange experience, which was really formative. And I came back and I've decided that actually it was the lab and not the clinical work that I'm really excited about at that point. I remember thinking that in a lab you're actually better if you doubt things, and actually in the world it wasn't really a great idea to start doubting what you were giving patients or what you were sort of and that to me resonated in sort of, but I still had very early on an interest in sort of applying research to clinical impact. And I did my PhD still in Milan. It was an English PhD with a sponsoring establishment from the European Institute of Oncology. And then I moved to London and I started my first independent projects, first in UCL, then jointly funded by University of Cambridge and a small biotech, and then back in UCL at the Cancer Institute. And in 2013, I've joined King's. But so these are the people actually that were really mentoring me through the steps. And it's very important to surround yourself from people you actually trust and you think are aligned with what you want to achieve. I think your kind of leverage grows as your career grows, but I think very early on it's good to check whether what you're doing makes you happy or it doesn't. One of the elements that was starting to really play as I was writing my papers as every academic was that I was very interested in the biotech space. And so sometimes if you observe what an academic lab does and what a biotech does, I was told by one of my mentors that you have to sort of think of the lyrics and the tune to the same song. So the languages are very, very different. The understanding is different. The kind of impact value is different. So you have a publication in one sense, you make knowledge in academia, but you want to really make a drug, for example, in a biotech. And so sometimes there's also something I call cross arrogance between the two fields, which has really been something that I wanted to sort of try to take down in for as little as I could. But I think we also have to sort of think that scientists are very different people, and they're like, there's an image of science that's been changing a lot in recent years, and most of it for the good, I think. So this, for example, is a picture of the lab of Professor Fiona Watt, who's my line manager at King's. And this is one retreat from the lab. And I mean, we kind of used to have these, we didn't do it last year for reasons you understand, but we used to have 3 days of sort of kind of lots of scientific discussion and lots of fun in the afternoons. And so I think you have to think of a scientist as a human being with quite some possibilities to have fun in the process. But what you actually hear, especially in academia, is that the path is very linear. So you learn, you make a discovery, which is sort of your PhD. So the definition of a PhD is sort of you advance the knowledge of humanity up to this stage in a little part. And then you apply this even more in an independent thinking. So you complete your training with your postdoctoral training, and then you become independent as a group leader or a PI. But this is actually quite a bit of a myth, because the reality looks a little bit more like this. So only 4% of PhD holders in the UK end up having a permanent position in academia. And there's a lot of changing into the environment of biotech. And now, for example, people like me have sort of come back and forth. And this used to be impossible a few decades ago. And also, there's a lot of careers outside science that PhD holders decide to undertake. So this is scary in a sense, because it's an extremely competitive environment, but I think it also gives a sense of flexibility in this new world, especially in this type of stuff I do. I actually now I've grown to value things with a different lens to the one that that kind of is based on publication or academic track record. And I think eclectic careers and kind of CVs that demonstrate drive and independence are very appealing to me at the moment, compared to very standard kind of high academic grading. And so what I'm trying to convey is that I think the path can be, if there is anything linear in the path, you can sort of imagine that you can first focus on yourself, then start focusing on the actual dissection of what tasks are between, for example, you and your supervisor, and later on build leadership in sort of team building. And I will try to explain this better in the following slides. So what I do is based on imaging. And so we use software to analyze images from cells. And the times we're living are very rich in imaging, and actually the data and the images are kind of different faces of the same thing. So here, for example, you could see in the early 2020 a map of the UK flights on the island. And just based on the tracks from the planes, you can sort of figure out that there must be a big city in the south of this island, for example. And so we do very similar things with cells. Cells, of course, are the kind of single unit of life in our body. And in particular, we work with stem cells. And actually, the defining feature of stem cells are the capability to self-renew, so that to produce identical cells to themselves and to differentiate. And to simplify things, it's a little bit like having a family or having a career. So stem cells are like managing to sort of make copies of themselves, similarly to having a family, and sort of learn a path in which they specialize in and become a different type of cell. So these two are the defining features of stem cells. And typically we refer to cells that are in our body. So they maintain, for example, the skin layers. Many tissues have the possibility to be maintained by a resident population of adult tissue stem cells. But very early on, we define embryonic stem cells as the cells that immediately come after the zygote starts duplicating. So I think there is something very philosophical about the fact that from one single entity, very simple, you have all the information that gets you to a newborn. And basically, this idea that self-renewal and differentiation would happen kind of inexorably and has been completely challenged by findings that came from the 26 in mouse and 27 in human, and gave the Nobel Prize to Shinya Yamanaka and John Gurdon, which basically allowed scientists to take a very differentiated cell, so a cell that has already learned a career, a function, and bring it back to an embryonic-like stage. So this we refer to as induced pluripotent stem cells. Induced because they have been made pluripotent, and pluripotent because they can become different type of cells. In particular, different type of cells very similar to the embryonic ones. And so once you have the right cell type, you can think that the drug of the future, if you wish, is the possibility of using the cell as a therapeutic itself. And so you can transplant the cells and you can transplant tissues. Like bone marrow transplantation is the earliest example of cell therapy, if you wish. But now there's a lot of applications that are really beginning to be real in the clinics. And it's also important to know that through this process, such as induced pluripotency, you can obtain specific cell type that are required to be replenished in the body. But there are two very different type of situation in cell therapy. So you can decide to take the cells from one person and expand them and engineer them and put them back into the same person, which has big problems in terms of logistics and models. And it's unclear where the product is, if you wish. So you have to have a hospital that isolate the cells, and a lab that sort of engineers and expand them, then a hospital that puts them back in the very same patient. And so the other option is the possibility of using what's called allogeneic cells. So you take cells from one person, expand them massively, and then ultimately you end up having something that is very similar to the aspirin you buy in the pharmacy. But the problem with this approach is rejection, such as for transplants. If you take somebody else's cells and put them in your body, there are several problems in terms of rejection. And so the routes that are being explored are in very broad around these two lines, but also other people think that the stem cells could also be immediately used to obtain the right cell type on which you can test drugs and find drugs in drug discovery. And I will keep referring to this free figure of the sort of commercial, clinical, and academic, because I think that the cooperation of these elements in this type of research is very important. Okay, so let me bring you to an idea of how the day looks like, as I was asked to give you a sort of picture of what a day would look like. So we observe cells under the microscope, and this can be done in several ways. I mean, here you have two different types of cells, and you can see that very quickly that in one condition, the cells are expanding faster. We don't know if they replicate faster. For example, they could be replicating at the same level but dying less. But what we know is that they are expanding faster. And so this type of things can really give you a quick sense of chemicals, for example, that would make cells proliferate faster or slower, etc. In this particular case, these cells on the left are human-induced pluripotent stem cells human-induced pluripotent stem cells derived neuroepithelial stem cells exposed with one ROCK inhibitor, which is just a compound that makes them happy and proliferate faster. But basically, we can go much further and ask the computer to count for us. So here we are overlaying images such as the ones before, but we are also asking the computer, tell us when you find the cell. And so you can see blue objects when the computer finds a cell. You can see green objects occasionally when there is an erroneously segmented cell. And then when a cell kind of rounds up before division, you can see the nice red dot. So we can not only ask the computer how many cells are there in the plate, but you can also ask how many cells are replicating at this particular moment. Another thing we can do is tracking. So we can say, can you follow up the cells throughout time and tell us where they are in the position on the plate. So this is work done by Victoria Tsang, the PhD student in my team. And you can see in the bottom left a video of the cells moving, but on the bottom right, you can see the tracks that are forming that follow the cells. We can do, for example, videos of colonies of cells. So here you have cells that are growing on top of a layer of so-called feeders, which are mouse cells that are used to make them, to provide like an environment that is conducive of their proliferation. And the computer, again, can help us kind of distinguishing the different layers. So we would have the nuclei of the colonies, and we can discard the layers of the feeders just based on how distant the nuclei are. We can do interesting things, for example, by following how a parameter of a cell, like the area, changes over time. And then we can look at taking away the time and sort of just comparing a cell that has grown faster or slower. And I won't go into details, but I'm happy to answer questions about this. This is useful when you want to kind of correlate genomic information with the imaging data. And then we can use artificial intelligence and, for example, ask whether the computer can help us distinguish conditions that make the cells grow in 3D as kind of spheres, like you see in the bottom left, or conditions that make the cells in 3D proliferate more as budding objects, like in the bottom right. And again, we're using tricks like subtracting 1 hour with the next, just to be able to get a clean image that can be segmented better. Inside the cells, we can color subcellular structures, like this is the mitotic spindle, which is this beautiful structure that helps the cells dividing. And also, we can look at different type of environment on which the cells reside, and then ask questions like, where are the nuclei, where is the cytoplasm? We can ask whether a cell is single or want to be with their mates, and we can get the numbers of in the parameter to sort of get a snapshot idea whether this is a good cell or a bad cell. And this is beginning to have applications also for cell therapy. And for example, we've done a big study where we've taken 75 people and 110 cell lines from them, and we've sort of distinguished the difference when the cells were exposed to different factors between the difference from person to person. So there is some sort of intrinsic machinery that makes a cell a little bit larger, a little bit more preferring to be single or in colonies, etc. And we can identify this type of signals and then look at the genome of the different people and see if we can find genes that are predictive of this. And so in the past, we've done this in person, which was great. And the Center of Stem Cells Regenerative Medicine at King's is very open to these kind of activities. So by all means, follow us and kind of get an opportunity if you're interested in seeing how a lab works. One particular facility we have set up is called the Stem Cell Hotel, because scientists come to us with their requests in terms of imaging, and we can offer some ideas and expertise, but also we can offer access to the devices that are required, which is a model that is being explored in different ways. And this is just to give you a sense of how kind of broad our interest goes in innovation. We have been working with a company that is in Shepherd's Bush, and sort of now in this particular climate last year, where we all were trying to be useful in one way or another, we've been looking at a way to take a shipping container and turn it into a modular laboratory for diagnostic. So we got some initial seed funding, and now we have a prototype in Jersey with OpenCell. And I think this type of things are really great in the sense that everybody, like the big scientific community, had to sort of come up with something a little bit different from their daily job to try and see if they could be of help in different ways. And now most of my time is spent really remote working, but I switch between my role at King's and this new adventure starting near Cambridge in a company called BitBio. And again, I'm happy to answer questions if you're interested in that. So now in the last part, I would like to leave you with some kind of useful tips. I was asked to share something that I wish I was told at the beginning of my journey. And so the first thing is about how you work on yourself. So there is this small movie that kind of I've taken from the same reference as the first slide, that basically kind of says that you start your journey, you start learning things a bit in a general way, and then you kind of embark in a journey, a little bit like a cell chooses their lineage. And at the end, when you are at the end of what's known by humanity, you sort of pierce out your PhD. And so you manage to sort of increase. And for you, this is a huge thing, but actually it's also quite humbling to see that in the big picture, this is how others would see your PhD, just a little expansion of the knowledge at that time. And the thing that I think emerges after you've worked on yourself is sort of how it's important to work with others. And I think initially you have one person you're relating with. So I think kind of very early on, thinking what it means to complete a task, what it means to sort of receive instructions. Do you have enough instructions to complete the task by yourself? optimize the times of the interaction. So meet, for example, once a week and sort of gather all the questions in one place. These are things that people don't really tell you, but go a long way if you do them right. And then later on, you begin to be kind of the center of a place where tasks get sent back and forth. And so I think not everybody needs to be a leader, but there are also different types of leadership. So I think sometimes people don't challenge themselves enough to sort of think very early that in sort of taking on them some tasks that require interaction with people. So I think that there is a bit of a myth of one single type of leader, but actually it's more about kind of making your own leadership skills and your own leadership style real. And so if I can put this back into the context of the beginning, I think if you want to follow a path, just start working on yourself, but also early on think about how you can work with one single person, and then as early as possible think about how you can interact with many people in a team. So just to conclude, I think these are the things I would recommend. And also there was this phrase that I've seen in my son's old master's office, which has really struck with me. So the person with the ability to describe reality without laying the blame will emerge as the person one can trust. And I like it because it's not judgmental on people who blame or not. It's just like a pragmatic thing that is very real. If you can, and it's a big thing, and occasionally life throw at us things that are tough to justify without laying any blame. But if you start trying to develop that ability, it will again go a long way. So in my personal path, the real niche was a bit of an epiphany around the end of the postdoc. So I sort of thought that I could easily spend the rest of my existence thinking the particular mechanism I was studying, which was exactly what academia required me to in a sort of very clear way. But I was very enthusiastic about the possibilities early on to use the type of mechanism to make drugs for glioblastoma, which is a brain tumor that we were working on. And also, even more, I was very interested in making platforms that could find other drugs. And it was very clear that the sort of path on the bottom left and the bottom right were not there. They were really something you had to look actively for. And I think that this moment was very interesting. And like, for example, now I have the privilege to really be part of a group from the Academy of Medical Science that is really focused into this triangulation of academic, clinical, and commercial activities, very broad, very different backgrounds, and incredibly good and supportive, and feels a bit like a class, really. And last thing, and then I'm open for question, is always check if you're aligned with the organization you're in. Always think of what makes you happy, and at the same time, just judge if you are engaging in the same path as the sort of organization you're in asks you to be, because it's very easy to get derailed. And I think in many cases it's better to try and be happy more than anything else. So just to conclude, these are the people who have done a lot of the work I've shown, and members of the group at King's. And I am happy to take questions.